Well, hey there, lab friends. So, I'm going to close the door. Why did I start before closing the door? I don't know. All right, so we're picking up with part 2 of our experiment. If you recall, last week on molecular and cell biology, we isolated plasmid. Some of us made lots of plasmid. Some of us made less plasmid. No judging, at least not out loud. What we're going to do today is put it back into bacteria. This is a classic molecular genetics technique that is known as transformation. Now, just to provide a bit of historical background, the term transformation comes from an experiment done by Oswald Avery and Griffith and MacLeod, in which in the 1930s, 1940s, the idea that DNA was in fact the genetic material was not yet really fully understood. Chromosomes were understood to be the basis of inheritance, what's known as the chromosomal basis of inheritance. And this had been carried out by Thomas Hunt Morgan using fruit flies, in which he could actually distinguish chromosome identity because flies in their salivary glands have really big, what are known as polytene chromosomes. And you can outline and kind of the bumps and the compactness and spreading, the bands. You can identify the individual chromosomes. He was able to recognize that a trait was associated with a specific chromosome. So we had the chromosomal basis of inheritance, but exactly what was the genetic material was not yet known. I mean, if you think about this, back in the turn of the 20th century, we knew a fair amount about proteins and enzyme kinetics and enzymatics. And so one would expect that, probably proteins were actually somehow transferring this information, this genetic information. DNA at the time was about as interesting as cornstarch. I mean, it's a negatively charged, homogeneously negatively charged molecule. It doesn't seem like it has the ability to carry information. Well, what Avery began to do was look for some experiments in which he could begin to assay whether DNA or protein were actually carrying the genetic information. He took advantage of the fact that there are 2 different strains of a pneumonia-causing bacteria. It's a Streptococcus pneumoniae, that there is a lethal strain and a non-lethal strain. Turns out the lethal strain is known as the S strain. The actual truth is that the non-lethal is referred to as the R strain. Well, what he didn't know is it turns out that the R strain is missing part of the metabolism or part of the metabolic pathway to build the candy coating capsule that is in fact an exotoxin that causes pneumonia. And so the R strain is named for the roughened appearance of the bacterium. So it's kind of bumpily looking. And the smooth strain, like an M&M, has got a really slick candy coating. All right, so what he found was if you inject S strain bacteria into mice, they die. If you inject R strain bacteria into mice, they get up, they walk away, they say, what the heck? So what he began to do was crack open S strain bacteria and mix them with R strain bacteria, and he observed something that he called transformation. That is, S, let's just call it stuff, because he doesn't know what it is yet, but S stuff, it could be protein, it could be lipid, who knows, has the ability, this marker is dying, has the ability to transform an R strain into the lethal S strain. And so I think actually Avery was the one that discovered this transformation, and he called this mysterious substance the transforming element. what is the transforming element? And that's where Griffith and McLeod come in. What they began to do was take apart the stuff that you get when you crack open a bacteria. And they carried out some fairly primitive but significant biochemical assays in which they used first like a lipase to break down all the lipid component. You still get S transformation. They used a protease. You still get transformation. They finally used DNase, the enzyme that degrades DNA, and no transformation. Now, what this says is, there's something to this DNA junk. DNA is, in fact, the transforming factor. So, because of that, we call this transformation, the adding of DNA to bacteria cells. Just as a little point, when you add DNA to eukaryotic cells, it's called a transfection. Why they made the change, I don't know. So, we're going to do a transformation. One of the things that is critical in order to carry out a transformation is the cells that are going to take the DNA up have to be treated in a very specific way. They have to be rendered what Avery called competent. So we're going to make competent E. coli cells. And there are a number of ways you can make cells competent. The standard, simplest way is to treat them for about 15 minutes on ice with calcium chloride. So what we're going to do is use a calcium chloride solution, about 250 millimolar, the exact numbers, don't worry about it, and add bacteria to it. Now, as we'll see as we talk, what we're going for is the highest transformation efficiency. The microgram, the number of colonies per microgram. That is our number. The more colonies you get, the better you are. The more colonies you get depends on how well competent your cells are. So, there are a lot of different ways to make competent cells. And ideally, the best way is to take a culture, grow it up overnight, then take and dilute that back into fresh medium, and you allow the bacteria to get into the mid-log phase. They're not too crunchy, they haven't built really heavy walls. And at mid-log, you then spin them down, keeping them completely on ice cold. Add calcium chloride for a few moments, spin them down a few times, and wash them in calcium chloride solution, and that makes them competent. They're fairly delicate when they've been treated with calcium chloride. They start to get some holes punched into them. So we're not going to do that. We're going to do a simplified procedure in which we take a plate of bacteria, and allow me to introduce you to my favorite bacteria strain, DH5 alpha. DH5 alpha, meet everyone. DH5 alpha is a phenomenally engineered strain of bacteria that allows for lots of easy manipulation of DNA. What you're going to do is take a loop like this and approach your plate thusly. Say, hello, plate, and physically scrape up bacteria until you get a wad of bacteria on your loop. See? And then you will have a tube of calcium chloride. These are twist-offs. You drop it in there, and then using your thumb and forefinger, go spin it betwixt your fingers to dislodge that bacteria. You can then use a micropipette to pipette up and down. You don't want to beat them too hard, but you do want to disperse them so they're not like a clump. There's a bit of a clump in there, but this is demo purposes only. All right, so then, let's just go ahead and wrap this up. We take one loopful of E. coli, and everybody will have their own plates. And you're going to resuspend in calcium chloride. We'll put it on ice, for 15 minutes or so. a critical thing you have to do is something called a heat shock. We're going to take them directly from the ice and put them into a water bath set at 42 degrees. I'll just tell you, 42 degrees, this is a lovely bath temperature, but that change in temperature causes the DNA to get taken up by the cells. Once we have heat shocked them, and we typically do this for, between 45 and 90 seconds of heat shock, we're going to allow the cells to recover. And so we'll add 1 mil of a media that is known as luria broth, or in the trade as LB. And we'll then incubate this at 37 degrees. How long will we incubate it for? Depends on how long you feel like incubating it. I would say a minimum of 15 minutes or so. It depends on how impatient I get. You want to incubate them for a little while. More than an hour is ridiculous, and we're not going to take that kind of time. And then we're going to plate them onto plates. Now, here is the key. The plasmid, as you recall, has the gene that confers antibiotic resistance. So we're going to, in fact, transform these bacteria from being sensitive to ampicillin to being resistant to ampicillin. And so the AMP resistance gene, known in the biz as AMP-R, this is the enzyme beta-lactamase. Beta-lactamase is able to break apart and break down the antibiotic. And the bacteria that are carrying our plasmid can now grow in the presence of ampicillin. So after the 15-minute, 37-degree incubation, the recovery, we're going to plate the cells. And I have, back in the back, several stacks of plates that are LB plus agar plus 100 micrograms per mil ampicillin. And we're going to use these guys to select for those bacteria that took up the plasmid. So we'll grow them overnight, and then we will count. Like Sesame Street, the count. You'll count the number of bacteria you got, and we'll do a little bit of calculations to figure out what is the colony number per microgram. That is your transformation efficiency. And I have a faculty meeting in 5 minutes, so I can't go into all the details and some of the fun trivia bits, but we'll talk about that in lab. If you get above 10 to the 5th, well, you're a badass, let's face it. That's pretty good for this level. We've got cells that you can buy from the man, or if you're very careful, you can make your own competent cells, and you can probably be getting 10 to the 6 colonies per microgram. We may, depending, break out the electroporator, which is a device that gives the bacteria cells a shock. You have to wash the bacteria extensively in water to remove all the salts. That's how you make them competent. You really don't do anything to it. And you put them in a little cuvette that blasts them with a big blast of electricity, and that allows them to take up DNA at a really high efficiency. And you probably can get anywhere from 10 to the 8th, 10 to the 9th, 10 to the 10th colonies per microgram. So it's one of the things with this efficiency is that you can have the same amount of DNA, and with different bacteria cells, my hair is uneven, with different bacteria cells treated in different ways, you can get vastly different numbers of colonies. All right, that's what we're going to do tomorrow. It's actually a lot of fun. I hope you enjoy it, and we'll talk more, and we'll have some time actually during these incubations and things, and I'll catch up with you tomorrow. Rock on.